Evening, my dear brothers and sisters. We have great pleasure in welcoming you all for a very interesting webinar today on COVID. Today, the speaker is a very eminent personality who has done a lot of work in COVID, a great academician, a great researcher, Padma Shri Professor Dr. Randeep Guleria. He needs no introduction, but still, as a formality, I would like to introduce him. He's the director of the All India Institute of Medical Sciences. He's a professor and head of the Department of Pulmonary Medicine and Sleep Disorders, member of the National Task Force for the Government of India for COVID-19, member of the Empowered Group 2 created by Prime Minister's Office in Multi-Sectorial Management of COVID. He's the chairperson of Clinical Research Group of Indian Council of Medical Research for COVID, recipient of Padma Shri in 2015, BC Roy Awardee, Doctor of Science, Times of Healthcare Achiever Award in Legends the category 2017, Director of the Decade by Indian Medical Association, awarded the Lung India Award for the Best Original Article in Lung India, received Association of Physicians of India EMERC Award for chest disease, chair and member of various prestigious national and international bodies, member of different national bodies to formulate guidelines for Indian management of asthma and chronic obstructive airway disease. The World Health Organization has sponsored this. Member of Oversight Committee of MCI, 460 publications, more than 350 invited lectures at national and international forum. He is the right person to talk to us. People have a lot of questions about COVID, especially this time, whether COVID has gone, whether we have developed immunity, innate herd immunity, whether the vaccines are going to come. I think Padma Shri Professor Randeep Guleria will explain all this to him. Please welcome our great Dr. Padma Shri Randeep Guleria. Sir, over to you, sir, for your presentation.
Thank you very much. At the onset, I'd like to thank Dr. Muruganathan and the American College of Physicians India chapter for inviting me for giving this talk. And I'll be talking based on two aspects. One aspect is the respiratory complications of COVID-19 and also on the future of COVID-19 vaccines. Before I start, I just want to know that my slides are visible.
Yes, sir. Your slides are visible, sir.
Okay, so when we look at, so I'll first focus on COVID-19 vaccine because that seems to be something that one is very much interested in. Vaccines have already been rolled out in many countries, and one would like to know when we could have that in our country itself. Now, vaccines are considered the most promising approach for controlling COVID-19 pandemic, and vaccines have actually changed how public health is being managed for over a century. Many diseases, infectious diseases like smallpox, polio, have been eradicated because of the vaccine strategy. So I think we must understand that vaccines have played a crucial role as far as mankind is concerned in eradicating diseases and in prolongation of life. As far as COVID-19 is concerned, there are over 50 candidate vaccines which are in human trials, and there are over 150, some people have actually now said it's more than 200, that are in preclinical trials. We have various platforms which are available as far as vaccine are concerned. Broadly, we have 6 platforms. And whenever we look at a vaccine, we are looking at 3 broad categories. One is the antigen. What is the antigen that you're going to use to cause an antibody response and get good neutralizing antibodies as far as the host is concerned. Then you look at a vaccine platform by which you deliver the vaccine. And then thirdly, you look at what is the route and how many regimens or doses of vaccine do we need to give. So I think these are the important issues that come up when we start looking at a vaccine. If we look at the vaccine platform that we have right now, we have what we call inactivated virus vaccine, which is the traditional platform that we have. We have now the new nucleic acid based platform, the recombinant viral vector platform, protein subunit, small number of vaccines and the live attenuated virus, and then the VLP, the virus-like particles. So there are a number of platforms which are being used, and as you can see, a large number of vaccines are in preclinical and clinical development. So if you look at the global landscape of COVID-19, and this is something remarkable, what has been achieved this year is unbelievable, that for one infection, we have now 203 candidates in preclinical stage, 58 candidates in clinical trial, and not one, but many platforms are being used to form a vaccine for COVID-19. This is something which shows how much technology and research has advanced, and also has shown the capacity that the world has developed of collaborative research which can be done across the globe between various institutes which have the capacity to really complement each other as far as vaccine research is concerned. Indian landscape for COVID-19 vaccine is also very, very encouraging. We have now almost 8 vaccine candidates which are at different stages, and many of them have now entered clinical stage. We have the Covishield, which is the Chadox or the AstraZeneca vaccine. The Serum Institute of India is now has reached phase 3, and phase 3 is more or less over, and they are now waiting for regulatory approval. Covaxin, which is the Bharat Biotech inactivated vaccine, which is with the ICMR. This is undergoing the final part of stage phase 3. The vaccine, which is by Cadila Healthcare, the Zycovid, which is a DNA-based vaccine, which is with the Department of Biotechnology, and this is in phase 2. We have also the Dr. Reddy's Laboratory vaccine, which is the Sputnik 5, which is with the Russian vaccine. And this is in phase 2 and phase 3 studies, which have now just commenced. And then Serum Institute of India is looking at a new vaccine candidate with Novavax. And then there are other candidates, the recombinant protein antigen based vaccine, the HGCO19 mRNA based vaccine, and the inactivated rabies vector platform vaccine, which is also being looked at. So remarkable R&D is being done in our country. Our country was basically known predominantly for manufacturing vaccines, but now we have really moved on into R&D as far as vaccine is concerned. So let's quickly get a feel of what these vaccines are. The nucleic acid vaccines are something which is new. These are the new type of vaccine platform that is being used. It's not new for COVID-19. These platforms have been tried in the past, but this is the first time that it is used clinical use. This platform was also being used for the Ebola virus vaccine, but did not go on to get or did not reach far enough to get regulatory approval. These vaccines use fragments of messenger RNA or DNA to produce the adaptive immune response through the host cells, and this happens by producing copies of the target antigen. So the mRNA vaccine actually induces the host cell to use spike protein, and this spike protein then leads to antibody production, and because it causes protein production, the antibody response is very robust, and this elicits both an antibody and a cytotoxic T cell response. The advantage of these vaccines, which we've seen over the last few months, is that it can be scaled up very rapidly and it can be produced quickly. So manufacturing and scaling up is something that this can be done with these vaccines very well. But these are relatively expensive, and booster doses are needed to really have a good immune response. Most of the data that we have from the Moderna and the Pfizer vaccine suggests that the efficacy is more than 90%, and various vaccines are being developed looking at the nucleic acid platform. Then we have the viral vector platform, which uses a viral vector to really as a platform. And this uses modified non-coronaviruses like the adenovirus, the vesicular stomatitis virus, which express SARS-CoV-2 spike protein and thereby produces the immune response. The adenovirus, which is being used in some of them, like the AstraZeneca, is not the human adenovirus, because the adenovirus, which is human, would therefore have already antibodies present and would not allow the propagation of the vaccine or the antibody production. And therefore, the Chadox vaccine, which is by AstraZeneca, uses the chimpanzee adenovirus as a viral vector. Again, this elicits both an antibody and a cytotoxic T lymphocyte response. There are potential safety concerns in immunocompromised patients, but the current data which is available suggests both from the AstraZeneca group and from others suggest that this is actually very safe. And host immunity to the viral vector may reduce vaccine efficacy depending upon the viral vectors that's being used. And like I mentioned, for the AstraZeneca, it's the chimpanzee viral vector which is therefore being used because we have no inherent immunity to that vector. And again, this can be produced quickly. Currently, 2 doses are required, but there is studies being done, especially by J&J, Johnson and Johnson's, to look at whether this can be a single dose can vaccine can also be given. Then we have the classical inactivated vaccine. These are the vaccine which are used commonly when we talk of influenza, polio, or hepatitis. And these use the killed version of the virus to generate immunity. They elicit neutralizing antibody, but don't elicit that much of a cell-mediated response. Can be safely given to immunocompromised patients. This is an inactivated vaccine, and therefore there is no concern as far as this is concerned. And this is a standard proven vaccine technology which has been used in several diseases like hepatitis, influenza, polio, and rabies. But two things are required. One is very often you require what we call an adjuvant to really get a good immune response. And many of these inactivated vaccines use alum as an adjuvant, and a booster dose is usually needed to get a good booster. And there are various vaccines which are being tried as far as the inactivated vaccine is concerned. Protein-based vaccines is also something which is being now looked at. This uses the protein of the virus to incite an immune response, and this uses recombinant viral proteins to induce the immune response. Again, it elicits neutralizing antibody, but again, the cell-mediated response is not that strong, and it can be given safely to immunocompromised patients. This technology also has been used in the past for hepatitis B, for pertussis, HPV, and the herpes zoster vaccine. Booster doses are usually needed for this. And again, a large number of vaccine candidates from Sanofi, GSK, the Novavax, which Serum Institute is also trying, are also using this technology as far as vaccines are concerned. Now, when we look at vaccine development, usually takes multiple years. More than a decade is usually required for a development of vaccine. But currently, as far as COVID-19 is concerned, this has happened in months rather than decades. And this is because a lot of things have been done where technology has been used and we have done things in parallel. So as you can see on the top, the classical vaccine program looks at first a lot of baseline work which looks at developing, looking at a target. The target is the antigen which can incite a good immune response. And very often we look at various targets in the virus which may be useful. Then we develop a development of partner selection, and basic preclinical animal studies are done to see whether this target does incite a good immune response, whether it's safe, and the safety signal in small animals and then in larger animals is good. And then after that, one looks at all the data, analyzes it, and then goes into phase 1 trial, which is the trial which does looks at dose response, basic data, and also tries to look at safety signals. Then we go into phase 2 trials, which also look at the efficacy and safety again. And once these data is robust enough, that is, the vaccine is safe, efficacious, and we know what is the dose that will give the maximum response, we move on to phase 3 trial, where a large number of individuals running into thousands are vaccinated in a randomized controlled trial basis to see for the efficacy of the vaccine. And then this data is presented to the licensing authority, the regulatory authority, to get the license. For COVID-19, this, because of the urgency and the need to develop a vaccine quickly, all of this was compressed, done in parallel, but without compromising on safety and efficacy. So the initial target ID was developed, a lot of investment was done, and this decision was taken at various government and industry levels to look at multiple targets and move them forward by looking at whatever data was available, both in safety and efficacy. So this was basically what was known as go or no-go decision to invest in the candidate. Once this was done, then in sequence, both phase 1, phase 2 trials were done, looking at safety, dose selection, efficacy, and also at the same time, clinical trials were planned and regulatory approvals were taken. And while this was being done, once we got good signals from phase 1 and phase 2 studies, manufacturing was started up with a lot of investment being done here, which was a risk, because if the phase 1 and phase 2 studies showed that the vaccine was not efficacious or that the vaccine had significant side effects, all this investment in manufacturing, development, scale up would have gone to waste. But that risk was taken, and manufacturing was done at this, simultaneously while phase two studies and phase three studies were going on. And this has allowed us to really cut short the time. And therefore, within a very short span of time, in less than a year, we have multiple vaccines which are available for COVID 19. So if you look at the landscape of COVID-19 as far as 10th December is concerned, there are a large number of vaccines which are now in phase 1, phase 2 and phase 3 trials. We have the Bharat Biotech Covaxin, which is now requiring 2 doses on day 0 and day 28. We have the Chadox vaccine, which is Covishield, which is University of Oxford, AstraZeneca and Serum Institute of India vaccine. Then we have other vaccine, the Sputnik, which is the Dr. Reddy's laboratory vaccine, the Covaxin, Covax, which is the Novavax, which is still undergoing phase 1 and phase 2 studies. And then we have various other vaccine, for example, which have been approved, the Pfizer vaccine, which has also been approved. The Cadila Healthcare vaccine is also undergoing trial, and we have again a new vaccine candidate which is being also looked at by the Serum Institute of India in collaboration with Codagenix, which is a company which is also looking at another vaccine candidate. So then this is basically a live attenuated virus which can also lead to a good amount of what we call mucosal immunity also. So we have a number of vaccine candidates, and what is very, very heartening for us as clinicians and researchers is to see that India has actually moved on to the next step from just manufacturing to doing a lot of R&D and research in developing vaccines. And these research platforms will be very useful for future vaccine development also. So we have now the AstraZeneca vaccine, which is waiting for regulatory approval. The Zydus is undergoing trials, and the Bharat Biotech vaccine also should be getting regulatory approvals soon, early next year. And that will give us a number of vaccines that we can give to our population. So there are a number of questions which come up with people ask, why do COVID-19 vaccine trial studies? No, we do, what are the studies that we do? And like I said, we do preclinical studies on animals, primates to assess the safety and immunogenicity. And then we do a phase 1 trial, which does a dose ranging and safety study in about 100 volunteers. In COVID vaccine, in some cases, it was done in almost 200 to 300 volunteers. And then a phase 2 trial is done to study the immunogenicity and safety in several hundred participants. And then we go on to the phase 3 trial to assess the efficacy of the vaccine in field conditions. And here the number of individuals runs in 20 to 30,000 or even more, so as to pick up any signals in the community which may suggest that the vaccine may have some side effects. Now, most of the vaccines which are coming into the market have gone through all of these robust and aggressive patterns to make sure that the vaccine is safe. What do we mean by efficacy? And this is something which has been put up by the US FDA and the WHO, that how do we look at vaccine efficacy? This is by a simple formula. And what WHO and the US FDA has said, that minimal efficacy criteria for licensure should be a vaccine efficacy should be 50% or more. And looking at the confidence interval, this means that the lower limit of the confidence interval should be more than 30%. We're very lucky that most of our vaccines that we are having have an efficacy which is usually much more than 50%, some of them of the tune of 70%, but many having an efficacy of more than 90%. So these are trials which were done. This is the Chadox vaccine, the one by AstraZeneca, and this is a paper which was published this month, which looked at the data from Brazil, South Africa, and the United Kingdom. And this, like I said, is a non-replicating adenoviral vector vaccine, 2 doses, 28 days apart. And this looked at, the interim analysis looked at around 11,000 participants. And the interesting thing was that they found that, whether it was by chance, that those who received 2 standard doses, the vaccine efficacy was 62%. But in one area where the participant got half the dose in the first dose and the full dose subsequently, 28 days apart, the efficacy went up to 90%, suggesting that if you were to give half a dose initially, you will stimulate the immune response. And then if you give the full dose subsequently, you got a much stronger immune response. This was interesting, and this is being evaluated in much more detail, because this will mean that you could have a dose modification and more doses could be available to vaccinate more and more individuals. The overall efficacy in this paper is around 70%. 8% of individuals had fever, headache, and fatigue, and two patients had transverse myelitis, but it was thought that this was not related to the vaccine. Then we have the Pfizer vaccine, and this is again an mRNA vaccine which encodes the spike protein. Again, needs two doses 21 days apart, unlike the previous one, which needed two doses 28 days apart. And here, the phase 3 trial has been done on 35,000 individuals, and adverse effects have been mild in the form of, again, fever, fatigue, headache, and chills in about 16% of the vaccinated individuals. This vaccine has been able to shown that it provides 95% protection against symptomatic COVID-19. But the main drawback, as far as the Pfizer vaccine is concerned, is the limited requirement of storage and transport at minus 70 degrees centigrade. For many countries, to have a very large rollout of this vaccine, keeping this in mind, becomes very, very challenging and difficult to do, especially in smaller towns and rural part of the country. Moderna has also now got approval as far as the US is concerned. And again, this is an mRNA vaccine, which has again been, the studies have been done in more than 30,000 participants. And again, this showed an efficacy of around 94%. And this has also now got approval in the US. And this vaccine has been now, is again being rolled out, and two doses are required 28 days apart. The Russian vaccine, the Sputnik 5, again, there is some data, and again, this uses two different adenovirus vectors, and which is very fascinating. It uses AD26 for the first dose and AD5 for the second dose, 28 days apart. And again, the efficacy of whatever data we have is over 95% as far as this is concerned. But again, the data is limited. The Dr. Reddy's laboratory is now doing phase 3 studies in our country, and hopefully we will get more robust data as to how good this vaccine is. We also have the Chinese COVID vaccine, which is now being used and which has been used in China. Very limited data available in the literature as far as this vaccine is concerned, but this has been used now in many Middle Eastern countries, and it is being rolled out. The claim is that its efficacy is around 86%, but we need more data before we can say how effective this Chinese vaccine is. But many countries have started using this vaccine despite robust data being available in the global literature. This is one paper which looked at the spike protein nanoparticle vaccine, and this is basically looking at recombinant protein nanoparticles, which again target the trimeric spike glycoprotein. This is given intramuscularly, two doses 21 days apart, and there is data which suggests that this would be again be a vaccine which in the near future may turn out to be another candidate which may come into human use. This binds to the neutralizing, this has a high binding and neutralizing response compared to convalescent plasma. And again, as in the other vaccines, about 6% of participants did have some degree of systemic side effects like fever, headache, myalgias, and malaise. So what are the major concerns and future challenges? Most vaccines are injectable and they elicit a system immune response, while we don't get what we call a mucosal response in the upper respiratory tract. And that is why there is a lot of interest to see whether we can have a simple nasal live attenuated vaccine, which can be used as a nasal spray. And this will give local mucosal immunity also and will not require the intramuscular injection. There are clinical studies going on as far as this vaccine is concerned. There's a concern of what is known as the vaccine induced enhanced response as far as vaccination is concerned. And this was in the past reported with RSV vaccine and also with some other vaccines, that when you have an individual who is vaccinated, you don't get that robust or good neutralizing antibodies, or the antibodies formed are there, but they're not neutralizing antibodies. And when this person gets the infection, these antibodies are not able to control the infection, and sometimes they may actually promote the entry of the virus into the cell and lead to more severe infection. So this is something which was a major cause of concern, but both in animal studies and human trials, this enhanced response has not been seen. I could give you another example, which there has been a lot of concern with the dengue vaccine. And this has also been seen that in the dengue vaccine, sometimes it causes a more severe disease in people who have been vaccinated. So something like that. But fortunately, as far as COVID-19 is concerned, it has not been reported so far. Then the issue is the duration of protection from vaccine, the need for repeated vaccination. This is a very important and billion-dollar question, but I don't think we have data right now. We need to have a long-term data. Most vaccines will definitely give protection for at least 8 to 9 months. I think at least a year, maybe more, but we need to have more data because we don't have enough follow-up. Also, as we have more and more vaccine candidate coming up, it will be interesting to see which vaccine actually provides long-lasting immunity, but that will also be something that will come out in the near future. But that is going to be a challenge because continuing doing vaccine trials in the future may also become a little bit of a challenge. And I'll come to that. A diverse effect of vaccines in population whom vaccine has not been trialed. This is something that we need to keep in mind, and therefore these vaccines are currently not approved for young children or pregnant women because they have not been tried in them, and therefore currently the regulatory approval is not there for pregnancy and young children. There are trials which have now started in looking at these vaccines in children, and once we have data from that, we could say that they can be used in young children. Long-term adverse effect of COVID-19 vaccine, especially those employing novel platforms, currently are unknown, but there's very less likely that there would be some long-term adverse effect. There is a lot of concern about the mRNA vaccine because people said we are interfering with our genetic code, and this messenger RNA will enter our DNA and change our basic DNA. But it's important to understand that this messenger RNA enters the cell but does not enter the nucleus. Secondly, this gets degraded very quickly within minutes of causing the response. And therefore, it's very unlikely that mRNA vaccines will have any effect as far as our DNA or genetic material is concerned. Once effective COVID-19 vaccines are available and have been approved, it will be logistically and ethically challenging to conduct placebo-controlled trials. And this is what I was mentioning, that once we have a vaccine which is available, will it be right to have a study where we have a placebo-controlled arm? May not be possible, and therefore what we will have to subsequently look at is to do what is known as non-inferiority trials, or we will have to define correlates of protection. How can we say that vaccine A is better than vaccine B, or it gives more long-lasting immunity? And therefore, we will have to have data which can identify association between antibody titer and vaccine efficacy. It may also be possible to license vaccine based on achievement on serological benchmarks rather than doing large-scale trials because it will have ethical issues. So this is another issue which will come up in the next year. A challenge is vaccine delivery and distribution, and vaccination and not vaccines prevent infection and saves life. And therefore, we are seeing vaccine hesitancy in many countries, and this is something that we need to address. There is a whole area of infodemic that is going on as far as COVID-19 is concerned, and concern about the vaccine, its ingredients, and whether it has something religious issues with that. I think we need to really start having good communication with the population. A lot of reassurance is required, and that is something which is very, very important. Who should receive the vaccine? There is a whole criteria and priority list which is there as far as the government of India is concerned, and I'll come to that. Who are the high priority group for vaccination? Again, I shall come to that. And we need to have a cold chain and a process in place, which is also being done very, very aggressively. So if you look at what the government of India has done, the government of India created what is known as the NEGVAC, the National Expert Group on Vaccination Administration for COVID-19. This is constituted on the 7th of August with the member Health Niti Aayog as chair and Secretary Health and Family Welfare as co-chair. I'm also a member of this group, and there are relevant representation of relevant ministries from state government. And we have meetings, we've had more than 10 meetings which look at various issues like prioritizing of population, procurement and inventory management, vaccine selection, vaccine delivery and tracking mechanism. So the guidelines, the vaccine strategy is being formulated as per the NECVAC guidelines, and most vaccines follow two-dose schedule. Like I said, there are some vaccine which may have a single dose or a three-dose schedule is also there in one vaccine, but that will be looked at subsequently. Currently, both the vaccines will have that are coming in India will have a two-dose schedule. Important to remember that vaccines are developed using multiple platforms and these are not interchangeable. Therefore, the person who's getting the vaccine must complete vaccination schedule using the same vaccine product. We can't jump vaccines right now because there is no data as far as this is concerned. Storage temperature for most vaccine will be as per the universal immunization program that the government of India runs for vaccination in children and in pregnant women, which is around 5 to 6 crore per year. And this is following 2 to 8 degrees centigrade. And rollout of the vaccine will be done using an election model. This is the NECVAC plan. This is the phase one where we will vaccinate around 30 crore population, healthcare workers approximately 1 crore, frontline workers around 2 crore, and prioritized group, that is all individuals above the age of 50 years of age and less than 50 years with associated comorbidities. This will be approximately 26 crore. It's easy for us to have this more than 50 years of age. The group that is less than 50 years is something that is being looked at, and I am the chair of a committee where we are working on this. We have committees looking at renal diseases, cardiovascular diseases, diabetes, chronic respiratory diseases, disabilities, other miscellaneous things like sickle cell anemia, and we are forming a simple a grid chart of who should be prioritized. So if you have diabetes for more than 10 years, or if you have diabetes with comorbidities, then should you be in the priority list for vaccination? If you have COPD and you are on long-term oxygen therapy or ILD or long-term oxygen therapy, or if your FEV1 is less than 50%. So various criteria is being looked at by a group of experts in each domain. And hopefully within a few days, we will have a criteria available for people less than 50 years who have comorbidities, who should be prioritized to be vaccinated. There is now a digital platform which has been created known as Covet, which is basically winning over COVID. This is using the same platform that the government has of eWin, which is the Electronic Vaccine Intelligence Network. This will look at COVID-19 vaccine distribution. It will look at end-to-end system across the world. It's the first end-to-end system across the world. It will look at various other parameters and will be integrating the Aadhaar card or the UHID, and will have various other parameters, including monitoring individuals subsequently for any adverse effects. Also, it will look at cold chain points. There are 28,000 cold chain points. There will be temperature loggers, which are around 23,000. And then there will be a lot of training that will be done. So the NECVAC, which is the National Expert Group, will have state level representatives. It will have district level representatives and block level. And in this network is already in place. And in at least 4 states now, a dry run is being done to make sure that from the manufacturing site to the delivery site, the vaccine is delivered safely, the temperature is maintained, and the vaccinators are there and they know how to vaccinate individuals who get selected. And this digital platform is also being looked at. So this is just to give you the diagrammatic representation of the COVID-19 vaccine delivery system, looking at the CoWIN mobile app, the temperature logger, the cloud server, and also being able to collect data of the beneficiary, tracking, vaccine session management, and this will also get fed into this database, and you will have a dashboard which will help us as far as giving vaccine across the country is concerned. So this is my first part of the talk. I'll quickly now cover respiratory complications of COVID-19, which is the second part. This is an unknown journey, and long-term outcomes of patients who have recovered from COVID-19 related complications are something that we're now looking at. It's important to understand that this is a virus which attaches to the ACE2 receptors using the spike protein, and the portal of entry is the respiratory tract and the upper respiratory tract mucosa, and therefore the respiratory system is the one which gets affected, but it causes multiple extrapulmonary manifestation. The virus causes a direct cytotoxic effect. It also causes dysregulation of the renin-angiotensin-aldosterone system. It causes endothelial cell dysfunction, and it causes a cytokine release syndrome. And the systemic complications of COVID-19 now are very, very diffuse, they involve various organs. And today, although we're discussing changes in the lung, it's important for all of us to remember that we must look out for other organ involvement also, because that is well known to happen with COVID-19. This paper, which was published quite some time back, post-COVID-19 symptoms. So, after 2 months, uh, 60 days, only 12.6% COVID-19 related symptoms. Most common symptoms of fatigue, dyspnea, joint pains, chest pain, and cough. And this is something that we need to. The British Medical Society published in the BMJ a very nice long COVID management protocol, where they have described what should be done once you have a person with symptoms 3 or more weeks after COVID-19 onset. What should be evaluated? What are the management conclusions? What should you look for? And what are the red flag signs? As far as the respiratory disease is concerned, COVID-19 pandemic is caused by the SARS-CoV-2 virus. It's basically affects the respiratory symptom in most patients, and in severe patient, it leads to acute respiratory failure. It can affect, it can have important respiratory complications like ARDS, pulmonary thromboembolism, and pulmonary microvascular injury, diaphragmatic dysfunction, and weakness, especially those who are on a ventilator for a long period of time. Those who are on steroids and immunosuppressants like anti-IL-6 are prone to secondary bacterial pneumonia. Interestingly, this also causes what is known as silent hypoxia or happy hypoxemia, and in some patients, it can lead to pulmonary fibrosis. So it's important to understand why does this happen. So COVID-19 it affects the alveolar epithelium and leads to development of ARDS. It affects basically the alveolar type 2 cells which express ACE2 the receptors, and that is why, since it binds to ACE2 receptor, it causes maximum changes in the alveolar type 2 cell. Uh, Therefore, it affects surfactant production because of affecting these cells, and it also, the infected cells produce virons which infect adjacent epithelial cells, endothelial cells, and macrophages. So in this cartoon, you can see when you have an infection in the alveoli, the type 2 cells, this is the type 2 cell, this is the type 1 cell. The type 2 cell get affected because of the fact that they have the ACE2 receptors, and this leads to a large number of changes in the type 2 cell, which leads to apoptosis, death, and dysfunction. This leads to decreased surfactant, which leads to atelectasis and collapse of the alveoli, which leads to shunting. It leads to development of alveolar 2 progenitor cells decrease, and this also further leads to atelectasis, and there is inflammation, which leads to immune cell influx and capillary leakage. So when the lung starts recovering, it requires reepithelization, removal of hyaline membrane material, and regression of stromal cell and leukocyte accumulation. And this is something which is very, very important. If injury is very extensive and there are inadequate number of type 2 cells, then repopulation is affected by specialized progenitor cells, and this then leads to lung dysfunction and pulmonary fibrosis. Also, what happens is that there is, along with that, there is vascular endothelium. The vascular lining in the lung also gets damaged. The capillaries are inflamed, and this leads to endothelial cell dysfunction, which leads to alteration in platelet and complement amplification, which lead to increased thrombosis. So there is clotting in the micro vessels of the lung. There is also vasoconstriction, increased fibrosis, and of course, increased inflammation. So endothelial dysfunction, clotting, and capillary leak occurs as far as the vessels are concerned, the pulmonary capillaries are concerned, and the alveoli, the type 2 cells, also get affected. This is very well shown in various autopsy studies. Now, we are also doing limited autopsies in our COVID positive patients. We've done more than 50 limited autopsies, and we are finding a lot of changes in the lung, which are similar to what has been reported in the past. The lungs are congested, they're patchy, they look really very, very solid, as they look like hard lungs. And there's diffuse alveolar hemorrhage with hyaline membrane formation, alveolar type 2 cell hyperplasia is there, there's microvascular thrombi, lymphocytic inflammation, and there is proteinaceous material. Therefore, this has been shown in various studies in autopsy finding from Italy, from Germany, that these lungs look really almost solidified with a lot of thrombus formation and microthrombi in the vessels. This has also been shown by what is known as dual energy CT scans of the lung, which has demonstrated abnormal dilatation and tortuosity of small peripheral pulmonary arteries. And these are found near the area of the lung parenchyma, which is involved, which is mainly in the periphery. And these are areas which therefore have problems. This can lead to the loss of hypoxic vasoconstriction, unfavorable VQ matching, and exaggerated hypoxemia. So this is a dual energy CT of patients with COVID-19, which again shows that in areas which show this classical ground glassing, the vessels are looking prominent, and there is features to suggest microemboli or in these areas where there is ground glassing, and there are almost what we call wedge-shaped opacities occurring because of that. So, what COVID 19 does is, besides affecting the lung alveoli, it is also associated with venous thromboembolism and pulmonary capillary microthrombi. So, it causes vascular endothelitis, it causes platelet activation, and it also causes endothelial dysfunction and altered blood flow. So, it actually meets the Verco's trial as far as COVID 19 is concerned, and this therefore leads to multiple clot formations as far as the lungs are concerned. This has also been shown to need to increase pulmonary dead space due to microthrombus, and the ventilatory ratio, which is a measure of efficacy of ventilation and gives an estimate of dead space, is actually altered and is found to be abnormally high. And thereby, it causes problem as far as the lung is concerned. We also have what we call happy hypoxemia or silent hypoxemia, and this has been another speculation as to why does it happen. We've had patients. I remember one doctor, lady doctor, who had COVID-19. She was on home isolation. She had no symptoms, and she just decided to get her oxygen saturation looked by her oximeter, and she found it was in her 80s. And then we rushed her to the hospital. She needed high flow oxygen, high flow nasal cannula, but it was only subsequently when she became aware of her saturation that she started saying that she's feeling a little breathless. Before that, she did not know that her saturation was so low. So, what is the basic reason for that? One is, of course, the shunt physiology, perfusion of unventilated respiratory units due to disordered hypoxic vasoconstriction. There is VQ mismatch, and this all is thought to be associated with silent hypoxemia. There's also a theory which is the neural mechanism that the virus actually enters the CNS, and this causes invades the vascular endothelium, leading to increased permeability in the blood-brain barrier and causes neuronal damage in the corticolimbal network, and therefore the perception of dyspnea is lost, and this affects the control of respiration. So this is a possible neurological mechanism which has also been put forward. But what are the long-term ramifications of these changes that I've just discussed? If we go back to SARS outbreak, which was in 2003, and there have been now studies which are a 15-year follow-up of outcome via CT scan. What they found was that in the early stage, there was pulmonary interstitial damage and functional decline, which was caused by the SARS outbreak, but this recovered gradually, and to a greater extent, recovery occurred within 2 years after rehabilitation. They did find some changes, but they found that in large number of patients, it was not progressive and was partially reversible. So these are important lessons for us when we look at what should we do in COVID-19. So long-term pulmonary manifestation have been extrapolated from findings of SARS pandemic. It's an ongoing area of research. And we know that older people and those with severe disease are at major risk factors. And other complications that we can see is chronic cough, bronchiectasis, and pulmonary vascular disease because of a thromboembolic complication. When we look at pulmonary function test abnormalities, and this is a study which looked at PFT and the related physiological characteristics in COVID-19 survival 3 months after recovery, and what it found was that in overall around 25% of patients had abnormalities in PFT, 16% had DLCO impairment, there was small airway dysfunction in about 7 and 12%, and the FEV1 and FVC was altered in 10%, and TLC, total lung capacity, was also altered in about 7% of patients. Radiologically, what we find is that there is this classical ground glassing, which is patchy, peripherally situated, which now all of us are recognized to pick up immediately, but it gradually progresses to more fibrotic changes, and the ground glassing disappears, but residual parenchymal bands or GGOs remain, and this is what is the post-COVID fibrosis. Sometimes you have changes which are more discrete, This is in another patient where you can see the dense fibrotic areas which are left at the base, so, and starting off with what was known as ground glassing, but then going on to dense fibrotic areas. This is another patient of ours where you have these dense ground glassing and then some degree of fibrotic with cystic lucencies which start happening as far as the bases are concerned. Another patient whom we had seen in the early part of the pandemic, and here again on follow up, what we find is that there is still some residual basal subpleural ground glassing with fibrotic areas, which is highlighted here, which we see as far as the post COVID sequelae. What are the risk factors for this? Is age more than 65 years of age, severe disease, length of ICU stay and mechanical ventilation, smoking and chronic alcoholism all have been looked at and found to be associated with higher chance of pulmonary fibrosis. How can we decrease this chance? One is to introduce the risk of ventilator induced lung injury by using classical ARDS network protected lung ventilation strategies. We should also educate or limit the exposure as far as our patients are concerned to environmental factors which can induce lung injury. Smoking cessation is very important, and these patients need to be followed up for quite some time. Drugs like pirfenidone and nintedanib are being used, but there's very limited data as to their utility. And like I said, as the experience from SARS told us that this is a non-progressive pulmonary fibrosis, and pirfenidone and nintedanib have been classically used in patients who have proliferative fibroblasts, ongoing inflammation, and therefore have been found to be useful. We need more studies before we can say that this can be standard of care as far as post-COVID lung fibrosis is concerned. Lung transplant, this has been looked at, especially those who have extensive fibrosis with respiratory failure. In my opinion, we shouldn't rush for it because we've seen the patients even recover after 2 months of having required HFNC. I have one patient currently admitted and another whom we had discharged some time back. The patient admitted was on high flow nasal cannula for more than 2 months, and he went into ICU psychosis because he was in ICU for 2 months. Now I've shifted him to the private ward. He is now on only 2 liters of oxygen, but he's developed very severe critical illness, polyneuropathy, and is undergoing extensive pulmonary rehabilitation of his especially upper and lower limb muscles. He can now just stand with the walker, but his oxygen requirement has dramatically come down. So many patients over 6 to 8 weeks will improve gradually and oxygen requirement will come down. However, we've had lung transplant. We've had now 2 lung transplant, one which was done in Chennai and one also has been done in Delhi of post-COVID lung fibrosis. So this may be done more and more often as we go ahead and start having more and more patients with end-stage lung disease. Follow-up CT, what should the rheological course of COVID-19, what is the rheological course of COVID-19 pneumonia? How frequently should we image them? And is the fibrosis permanent? These are often questions which are asked by everyone. If you look at data, this is a retrospective review of CT performed on the day of discharge and 2 and 4 weeks after discharge. In most patients, focal GGOs reduced and were only 9% from 17%. Multiple GGOs reduced from 80 to 23%. Consolidation were only 2%. Interlobular septal thickening also showed a decline. Subpleural lines decreased and irregular lines also decreased. So lung lesion of 25% patients were fully resolved in the first CT scan after discharge, and the rate of lung recovery increased to 65% after second follow-up. So that is what I'm stressing, that many of these patients will recover if we give the lung some time to heal and with supportive treatment. So the lung damage due to COVID-19 could be reversible. The optimum timing of follow-up scan is yet to be determined. We will actually wait to 6 to 8 weeks before getting the scan done because these patients already had many scans. It is usually prudent to wait for at least 6 weeks before we really consider detailed CT scanning. Finally, we have what is also known as a post-intensive care syndrome, the PICS, which has been also reported with severe COVID-19 complicated by respiratory failure and multi-organ failure resulting in ICU admission. This is defined as a new or worsening cognitive, psychiatric, or physical function after critical illness, needs extensive rehabilitation, both in the form of respiratory rehab and upper and lower limb and psychological support. So rehabilitation has become very, very important as far as severe COVID-19 is concerned. And this is what is I think forms the basis of the post-COVID clinics. that there is physical deconditioning and muscle weakness, there is fatigue, there's impaired lung function, there is what is known as brain fog and other cognitive impairment, and there is chronic fatigue syndrome, all of which we need to comprehensively address in our patients. And therefore, a multidisciplinary team approach is necessary when we look at rehabilitation of patients with post COVID sequelae. When do we call them for follow up? Normally, we follow the standard protocol that we use telemedicine to follow up for all our patients, especially who are sick. Usually, the first call is done sometimes on the second week, sometimes by the fourth week. And then, if need be, after the second and then the 6 months, if there's any symptom, the patient is actually asked to come for a physical checkup. And like I said, there has to be multi specialty follow up, whether it's pulmonary, cardiac, nephrology, neurology, or psychological support. So, to conclude, COVID 19 vaccine development is one of the great achievements of modern medicine. I think it's remarkable that one has been able to have so many vaccines available in such a short period of time. Equitable distribution of the COVID vaccine is imperative. If we have to get over this pandemic, the vaccine distribution has to be done in an equitable manner so that we are able to save lives and break the chain of transmission. As I've shown to you, we have a comprehensive plan to roll the vaccine program, and that is being worked on for the last few months, and a lot of detailing has been done in that. As far as post COVID sequelae is concerned, the degree of organ involvement, timing, and severity are variable with COVID. Post COVID sequelae is now being seen more and more commonly, especially now that we have a very high recovery rate, and we are seeing many patients who come after 2 or 3 weeks with many problems. They're not only respiratory but joint pains. Today, I saw a patient who had backache and also complained that his CT shows a score of 2 by 25, very minimal fibrosis. But he says that he's not able to breathe, he's not able to expand his chest, which suggests there is more of neuromuscular problem as far as his breathing is concerned. And many of these patients I've noticed have continued to have baseline tachycardia even for weeks. So this patient also, his pulse was 108, 110, and many patients continue to have that for weeks, which causes them to have palpitations and feel a sense of uneasiness. The lung can have various manifestation and some progress to severe fibrosis, and unfortunately, in a very small percentage, fortunately, in a very small percentage, there will maybe a need for lung transplant. But we need a regular multidisciplinary follow-up and screening protocol needs to be in place because this is a new emerging modality of treatment, and therefore we need to really be able to provide the best care for our patients. Thank you very much.
Thank you very much, sir, for that excellent, exhaustive, and very comprehensive, very clear presentation. In fact, I think your presentation has brought a lot of hope to all our medical fraternity and the community. There are a lot of questions, sir, but I think I'll ask very simple questions. When do you think the first vaccine will come to India, sir, in your opinion?
So, two vaccines, the Serum Institute's vaccine, the Bharat Biotech vaccine, have almost completed phase 3 trials. The data is being analyzed. Part of it has been given to the regulatory authorities. I think we're also waiting for the data from the UK as far as the AstraZeneca Serum Institute vaccine is concerned. I think by early next year, probably the first week of January, hopefully we should get the regulatory approvals. And as I've shown in my presentation, we have all the plan in place to roll out the vaccine. So as soon as the regulatory approval is there, within a very short span of time, we will start giving the vaccine to the priority group across the country.
So once the vaccine comes, suppose the 2-3 vaccines, like you said, Covaxin and other Reddy's vaccine, then Sputnik vaccine, if all three come together also, it's okay. You can distribute according to your that committee, which you said 30 crores of people you will cover. So hopefully 30 crores of people will be covered up to March 2021.
So depending on the number of doses available, and remember we have to give 2 doses. So if we are saying we'll cover 30 crore individuals, that means we need 60 crore vaccines. We have factored in actually around 65 crore because there has to be some degree which may get lost or may be misused, may not be properly used. Therefore, those many doses are required in the first phase. And in my mind, it will take at least 4 to 5 months for rolling out the vaccine to these 30 crore individuals and giving them 2 doses 28 days apart. But if we have sufficient vaccine doses, then we can start simultaneously vaccinating number of groups. It doesn't have to be that you do group 1, then you do group 2. It depends on the number of doses that you have. If you have sufficient doses, they can work in parallel. They can work in sequence, so in parallel, so that we can vaccinate elderly, frontline workers, healthcare workers simultaneously, and thereby being able to generate a sufficient number of people who are vaccinated to decrease our mortality and decrease the number of cases.
You were saying in your presentation, you give first dose off and then second dose full. There is a definite improvement. Are you planning that way, or what is your plan?
In India, the dosing that was done was both the first and the second dose was full dose. The regulatory approval in India will possibly in the first stage be for the first dose also being the full dose and the second dose also being the full dose. Later on, when we have more data, not only from India but globally, then the dosing modification may occur, and that is being studied in great detail by AstraZeneca, because that is what their observation was from the data they got from Brazil.
And suppose somebody has got first dose, can they move out of the house, or should be quarantined till the second dose is given?
So there's no need to quarantine yourself, but you must understand that your protection will occur normally 10 to 14 days after the second dose. If you get the first dose, your immune response is still not totally to the level to protect you. Some people feel that if I get the first dose, I get 50% protection. If I get the second dose, I will get 100% protection. It doesn't work like that. The first dose induces the immune system, and then subsequently, the second dose, like a booster dose, gives sufficient neutralizing antibodies. And after the second dose, it takes at least 10 to 14 days for the antibodies to be formed in sufficient amount to give you protection. So, COVID appropriate behavior has to be maintained throughout, because you are still prone to getting the infection.
Suppose, according to you, in the month of July or August, if you complete 30 crores, you think they'll develop herd immunity in the country?
I think so. I think if we are able to do this amount, if we look at the number of people who we have vaccinated, and also a number of people who may have got immunity because of infection, the serosurveillance data suggests that that varies from 20% to almost 40-50% in certain areas. Therefore, the mass of people who will have antibodies, either due to vaccination or because of infection, should be sufficient to say that to some degree of we would have got some degree of herd immunity.
The cost of the vaccine around in India, how much it will be?
So currently, the cost of the vaccine, as far as the program is concerned, that the government is planning, will be free of cost. The government, through various factors like CSR and others, will bear the cost. Subsequently, if it is out in the open market, I think the Serum Institute of Vaccine, the cost will vary from 300 rupees to maybe 700 rupees, but it is not going to be very expensive. The Pfizer vaccine, if it comes, is priced at a much higher dose in abroad, which is almost $20 to $30 per shot.
And once these vaccines are given, the antibodies will remain for at least 1 year, or what is your?
I feel that they should stay for at least a year. There are two important parts of this question. One part is, do we have enough data? I don't think we have enough data. Whatever extrapolated data we have from the trial suggests that the neutralizing antibodies and T cell immunity will stay for at least 1 year, maybe longer, but we need to have more data. The second part is, if this virus mutates, will the vaccine still be able to provide efficacy as far as the vaccine is concerned? Current data suggests that it will be able to provide protection. Whether the efficacy will change, no one really knows. And that only we will come to know in the coming months, because we don't know over the next few months what is the mutation the virus may have, because vaccination itself will also drive some degree of mutation, because the virus is trying to survive. So when it finds a lot of people are immune to its and not the virus itself will try to mutate to survive itself.
Because now everybody is scared about the new strain, virulent, and all that. Any idea what is your consolidating talk about that new virus, a new virulent virus strain?
So vaccine, the viruses are known to mutate, and we know that all respiratory viruses and other viruses mutate. We've seen this very frequently in influenza, and because the influenza vaccine mutates regularly, what is known as antigenic drift, we have a new vaccine every year. Now, as far as COVID-19 is concerned or the coronavirus is concerned, it does not really mutate that frequently. This mutation, which happened in was 17 different mutations were noticed, and some of them were at the spike protein, especially the N501Y, which was at the receptor binding domain. So this was a cause of concern. But this was noticed on the 20th of September. But what really caused an alarm is based on the epidemiological data. What the UK government saw was that in London and southeast part of England, the Kent area, where there was increase in number of cases, they also found that this correlated with the increase in the data suggesting this mutant circulating there more frequently in 60% of the population. They correlated this and showed that this was leading to more infectiousness. The R naught went up by zero point 0.4. R naught means how many people you can infect. One person can infect. If the R naught is 2, that means one person will infect 2 people. So this, if the R naught in the previous virus was 2, it went up to 2.4. And based on modeling data, then they said that this would make that this is 70% more infectious. They also looked at hospitalization and mortality and did not find any increase in hospitalization or mortality in that area. So what they concluded was this is a virus, this mutant is more infectious, it is spreading more quickly, especially in the younger population, but it is not causing more deaths, it's not causing more hospitalization.
They said 70% infectious, but only 9 or 18 amino acids out of 1,280 got mutated. So something very soothing news, because otherwise the media and everybody says virulent, virulent, and everybody's scared. And your talk has given some hope. And regarding the healthcare workers who are already COVID positive, will you give them a vaccine, or what is your stand?
There's been a lot of debate in our group as far as this is concerned. There are logistic challenge which are there from the point of view of immunity. Now we know that once you get infected, the immunity that you have is very variable. There are some people who have very little antibodies and they disappear quickly, and some have a very high antibody titer and they stay for a long time. So it's rather than taking a chance, I think it's better that we vaccinate everyone irrespective of whether he's had COVID infection in the past. Secondly, logistically, it's going to be very difficult because we know almost 30 to 40% of people have mild or asymptomatic disease and recover but never get tested. They only get picked up in serosurveillance. Now, if we have to pick all of these group up, that means for all of them, we will have to first do antibody testing, see who has antibodies, and then those who don't have antibodies, we start vaccinating them. This logistically will become more difficult. Therefore, I think both logistically and from the point of view of giving better protection to healthcare workers, all healthcare workers, irrespective of whether they've had mild or severe infection in the past, should be vaccinated.
I think there are a lot of questions on vaccine. I will stop vaccine questions. I'll go to only 2 questions. What do you think about monoclonal antibody? Do you think it will come to India? What Trump had taken and what is available in US? Is it going to be available in India also soon?
So monoclonal antibodies are being made by 2 companies, one which Trump had taken, Regeneron, and one by Eli Lilly. The data currently suggests that they're useful only in those who have mild illness before they require hospitalization or need oxygen therapy. As a matter of fact, it did not have any benefit in the group which went into hospitalization. So early disease, it may be useful because they will therefore provide passive immunity. However, this is something which will be very costly, and therefore I think it will have limited use as far as our country is concerned. It may come into the market at some point in time. Here, I'd just like to say one thing, that unlike the vaccine research on finding a good antiviral treatment for COVID-19 has not been that robust. We haven't had that much of success. And although we have a vaccine, we don't have a good antiviral drug. So a therapeutic armamentarium also, I think we need to have research to improve that. The monoclonal antibodies may have limited role. We need to find a good antiviral which can actually prevent viral replication as early as possible.
And what about baricitinib? Baricitinib they are using instead of steroids. It's quite safe and side effects are less. Your opinion on that?
So baricitinib is also a drug which, again, an immunomodulator, which is also being used as a sort of replacement for steroids if it can be given. It's an oral medication. It's been used. It's available in our country also. But I think we still need more data before we can say how useful it is. But baricitinib, based on the NEGM paper, does tend to suggest, it does tend to support that if it is given along with steroids, it may be useful and may have a better response as far as the inflammation is concerned.
And you have clearly said for the fibrosis, you are trying the drugs, but it's not useful. What about methylene blue? Do you think methylene blue is useful in preventing any lung problems?
So methylene blue, there is very little data. It has been used in certain centers. I've talked to doctors in various hospitals, and some of them have used methylene blue. They've not been able to come out with concrete data to show it's useful. Therefore, I have my reservation as to how useful it would be. We need to get good data before we can say that a form of therapy is actually going to make a big difference.
Last question. What about post-COVID? You think post-COVID will be a big problem now? Post-COVID in India?
Post-COVID sequelae, we are seeing more and more because our recovery rate is good. It will be a problem to some extent. I'm hopeful that most patients in 8 weeks' time will recover and may not require long-term treatment. But there will be about 5% of patients, but in our country, that number is also very large in absolute terms, who will require long-term care, whether it be rehabilitation, oxygen, or management, because many of them have a decrease in their cardiac function because of myocarditis. Some have had stroke, and some continue to have hypercoagulable state and venous or arterial thromboembolic complications. So they will need some degree of close follow-up.
And in your hospital institution, long COVID cases are there, like post-COVID, long COVID with autoimmune problems, etc.
Yes, yes, yes. We are seeing long COVID cases, and this is causing a main cause of concern because as our recovery rate improves, our concern is how many of these patients are we going to see, and we need to develop a comprehensive multidisciplinary treatment strategy to follow up all our patients with long COVID.
Dear friends, regarding your questions on vaccine, Dr. Randeep Gloria's presentation was so good. We are going to upload it in our website. Those of you who cannot follow, you can download the talk and then leisurely go through, because every vaccine, every aspect he has explained. So you can leisurely go through. All your questions will be answered if you again download and understand. So I think before we wind up, Randeep Gloria sir, you are concluding messages for all our viewers and for the whole country, because everybody is very, very anxious, and your words of wisdom and your hope will be a soothing factor and comforting factor for all of us. Your word of wisdom and conclusion, sir.
Thank you very much, Dr. Mugunathan. First, thank you for inviting me. First thing I'd like to say is that when we look at vaccine, we have achieved remarkable success as far as vaccine is concerned, not only globally, but as Indians. And this is a matter of pride for me and for all of us. But I would say that we now need to have confidence in our research and our scientists. And there is now vaccine hesitancy which is coming in. I think we must understand that vaccine is the only way out from this pandemic. And all of us should work as a team to see that everyone who needs to be vaccinated based on the prioritization gets vaccinated. We work as a team to push the vaccine program forward. This is the first time that we are looking at such a large-scale vaccination program as far as adults are concerned. Even in children, like I said, the children vaccination program is very limited. This is a huge task, and it's a challenge for us in the country to really implement that. Secondly, as far as long COVID and pulmonary fibrosis is concerned, we need to really follow this up more closely, not panic. Many patients who have scars in the lung or have changes, ground glassing, at the time of discharge do improve over the next 6 to 8 weeks, and therefore just good pulmonary rehabilitation, even yoga and supportive treatment helps them, and often on follow-up the scar disappear and the lung function improve to near normal. So don't panic if you have a person who on discharge has some changes on the CT scan. They will resolve. You must just follow up these patients more closely. A very small percentage may go on to extensive lung fibrosis, which will need treatment, as I've discussed in my presentation.
Thank you very much, sir. I know you had back-to-back 5-6 webinars. You had to be given interviews to so many televisions. We saw your interview in so many televisions, so busy. In spite of that, you took time and spent some time with us and gave good, hopeful messages. And new viewers, don't worry. Those who have you not have attended, please don't worry. We are going to upload SARS wonderful lecture in the website. You can download and leisurely you can go through. And the days to come, as he said, we'll have definitely COVID-free world, and the new normal will be a better healthy persons. And now we are geared up for any virus in the future. We can prepare vaccine in 45 days because the technology we have learned, especially mRNA technology, if we have learned, in 45 days, any virus comes, we can develop a vaccine. That is the good news. In spite of the COVID, we have got a lot of good news, and we'll be positive and we'll stay free. And as he rightly said, please follow the clue. Whatever I say, usually I say follow women. Women means wear mask, always observe physical distance, wash your hands, and take enough diet and exercise, and don't touch nose. Always say namaste. Namaste, sir. Thank you very much, sir. Thank you very much for that time, sir. And I thank all the viewers for your participation. And we'll keep posted all the questions. And in WhatsApp also, we are going to give the link. So those of you who are not, then you can send the link to everybody and then learn about the vaccines. Thank you, sir. Once again, thank you very much, sir. Good night, sir. Good night, sir. Thank you, sir. Thank you. Thank you, Nishant. Thank you.